Tutorial: Preservation of Human Stool Samples. In this tutorial, we will demonstrate how to preserve human stool samples in order to molecularly identify the different soil-transmitted helminths. It also enables molecular identification of single nucleotide polymorphisms in the beta-tubulin gene in these helminths associated with anthelminthic resistance. 1. Equipment. To preserve stool samples, the following equipment is necessary: 70% ethanol, a plastic beaker of at least 150 milliliters, falcon tubes of 15 milliliters with a screw cap, a scale, a pipette allowing one to transfer at least 5 milliliters, a stirring device such as a wooden stick, and a parafilm. 2. Procedure. We only want to preserve stool samples that contain helminth eggs. After this first selection, it is still possible that we are unable to preserve and store all these samples. Therefore, it is recommended that we make a new selection, either randomly or based on the fecal egg counts. Overall, the likelihood of having positive PCR results increases in function of increasing fecal egg counts. In this example, we have randomly selected 12 out of 24 stool samples that contain eggs of soil-transmitted helminths. To start the preservation procedure, label a falcon tube and place it on the scale. Press the tare key and add exactly 1 gram of stool. To avoid contamination of the stock of 70% ethanol, it is recommended that you start by transferring approximately 150 milliliters into a plastic beaker. With the pipette, add 5 milliliters of 70% ethanol to the tube and thoroughly homogenize the suspension with the stirring device. Afterwards, 70% ethanol is added to get a total volume of 10 milliliters. Thoroughly homogenize the suspension once more by shaking the tube. Finally, parafilm is put around the cap to avoid the evaporation of the ethanol during storage or shipment. The procedure can be repeated for the remaining stool samples. 3. Acknowledgements. The raw material for this tutorial was collected during a field study financially supported by an institutional university project of the Flemish Inter-University Council. The Research Foundation Flanders supported the development of the tutorial through a postdoctoral mandate grant.